Hey, someone's joined. Hello, Rose. All right, you two, you ready to go?
I just need to pull out my notes really quick and I will be ready.
Sure. Especially as there will be exam 3 this time next week. Isn't
Have
that
you
crazy?
figured have you figured out how you are going to do the exams?
I have. I just haven't had time to do it yet. So it's all going to be on Blackboard, um, similar to the homeworks and the like you've had already, but it will be a timed um, multiple choice exam on Blackboard.
Have you figured out how much time you're going to give us or
So it's obviously it's going to be open book, right?
uh-huh.
Because you know you're at home, so the time will be the same amount of time as you have in class for the exam.
Okay.
So if you really, really know your stuff and you know where everything is, it should be better for you.
Mhm.
If you don't, then you know it should be probably about the same as if you were doing it in class, to be honest. So hopefully the fact that you're not getting kind of in-person instruction will be balanced out a little bit by the exams being open book.
Yeah,
Uh,
I've seemed to notice that I almost seem to be studying more that I'm doing stuff at home
hmm,
because I'm having to pay more attention to everything, you know?
right. Yeah, yeah. I mean, and for some students, I think it's actually better this way round, because that frees up time for you to ask questions in this session.
Mhm.
For the less diligent students, I think it's far, far worse, you know, because they don't have that self-motivation to do the work even when no one's telling them they have to.
Mhm.
So it'll be interesting to see come the end of the semester. I don't think for the two of you it's particularly an issue, but if you have friends who are kind of marginal in various subjects, or if you are in other subjects, think hard about the credit, no credit option. Um, but personally, for the two of you, I don't think that you'd be best served by using that. I think you're both uh, at least heading for Bs, if not hopefully better. But there's certainly that option as well. Anyway, so what I think we should do first is, and Rose, you can unmute your audio if you want, unless you have like particularly noisy cats or something. Um, is just to field any questions about the transcription lecture now that you've all had the chance to go through that. If you have any particular questions, stuff you want to go back and cover, let's do that first, and then we can get on to the RNA processing and translation PowerPoint, which is pretty chunky.
I don't think I have any questions about the slide 15 if Rose does, if you want to get started there.
Rose, how about you?
Yeah, I had a question on slide 5 of the PowerPoint 15.
Cool.
And then
Hello,
I
Michael.
was just wondering about the side chain.
Uh-huh.
The R, right? It's similar to a nucleotide, so that would be like our nitrogenous base. So the side chain is our particular chemical structure of the amino acid that's being added, or
Correct.
Okay.
So, and just saying hi to Michael. Michael, you can unmute your audio if you want, and we're just going through PowerPoint 15, any extra questions, stuff like that.
Okay.
Yeah, so essentially, the analogous thing would be, like you said, the nitrogenous base in nucleotides where you have the A, C's, G's, T's, or U's. In amino acids, you have the R group, which is what this is. And so essentially, these are all of the amino acids, and the R groups are what are highlighted in color. So the bit that's shared between all amino acids is the backbone, like the phosphate sugar backbone in nucleotides. You have the amino carboxyl group backbone, right, which is, can you see what I'm doing on screen?
Mm-hmm.
Yeah, so that's this part here with the alpha carbon in the center. And then the R groups are the extra bit which essentially creates that specific amino acid.
Oh, okay.
So for glycine, it's just a hydrogen. For lysine, it's this long carbohydrate, not carbohydrate, carbon-hydrogen chain, and then an amino group at the end, right? So it varies. And actually, lysines are, hang on, this is a funny segue back. You remember, question, what charge are histones?
Negative. Positive.
Right, because DNA
Is negative.
okay. What's at the end of the lysine?
Hang on. I can't see it off of your screen.
Oh, okay.
Positive.
It's positive. So the lysines are the main amino acids that make histone tails positively charged.
Oh.
And those are the lysines that are methylated. No, not methylated, acetylated, sorry, to reduce the binding of DNA to the histones, essentially a new chromatin versus heterochromatin. So yeah, that's a neat kind of connect the dots kind of thing. Cool. Did that help?
Mm-hmm.
Michael, just give Michael a chance. Do you have any questions about PowerPoint 15 transcription?
Not right now.
Okay.
But yeah, I'll let you know.
Yeah, well, if you do, then just write them down, and either we can look at them on Thursday, because Thursday is actually billed as a recap session.
Okay.
So any questions you have about anything that we've covered up till the end of essentially this PowerPoint 16, write them down and then bring them to the meeting on Thursday.
Got you. Yes, sir.
Yeah, and then we'll be able to go over those in detail.
Sweet.
Okay. Anyone have any questions, more questions about 15?
I had a question really quick about the exam and what it's going to cover.
Uh-huh.
Is it going to cover, what is it, 13
Oh, you're talking about chapters, aren't you?
through? Yeah, like what is it going to cover here?
So I've just brought up the actually not a whole lot, to be honest. It's a fairly short well, I mean, it feels like a short section, right? So essentially it's these subjects here, which are let me see, 12 PowerPoints 12 through 16.
Okay.
Because we're going to hit is that someone's dog in the background?
I'm sorry, my dog is snoring.
That's why I have mine muted, because it doesn't like to be quiet.
That's really funny. Yeah, so after exam 3, so the last part of the semester will be gene expression regulation, a little bit of biotechnology and genomics, essentially. So that will be, where are we at? Essentially 17, PowerPoint 17 through 21. And my aim actually is to get these done this week as best as I'm able to. So I can free up time to get the exams done and my other class and other things I need to get going with. So essentially the format is going to be exactly the same as exam 1 and 2 in terms of question difficulty and you know, the kind of questions that I ask essentially. So just basic recall, right, which is where you should be able to, if you're quick, pick up the stuff through having open notes. Although, to be honest, open notes can be somewhat of a curse as well as a blessing, to be honest. But there'll also be several questions where you're going to be asked to figure stuff out, right? And you're already familiar with those from, you know, previous exams and the kind of questions that I give you in, or gave you in GenBio 1. So essentially it's going to be exactly the same, just doing it all online instead. I don't mind listening to your snoring dog, Alana. It's quite cute.
Oh, okay.
I thought someone was like really hungry or something. I was hearing their stomachs rumble. It's
no,
like,
she has a really short snout, so she snores really loud.
oh, that's very cute. Okay, so everybody ready to start? Have a quick run through the translation one.
That sounds good.
Yeah, anything that we don't get to today, it can just fall over into Thursdays, and we can finish it off there. Right. So there's two big things in this PowerPoint. It's actually a pretty bloody huge PowerPoint. If you look at it, it's 35 slides long, so it's pretty chunky. The first one is RNA processing, right? So if you look back at 15, where did we leave off? Here we go. So this is what a eukaryotic gene looks like, right? From 5' to 3'. So 5' UTR, exons, introns, 3' UTR. And when you make a transcript, that's what that transcript looks like. It's a direct copy, right? So everything you have in DNA is copied faithfully over into mRNA. Obviously, that's not going to work because it's only the green bits here which code for protein. And so everything in between has to be removed. Also, another big deal for mRNA is it's very fragile. It's very prone to degradation, where enzymes called RNases eat it up just very, very quickly. And that's actually part of the control of gene expression, which we're not really going to go into, but how long that RNA hangs around for is also part of how much protein is produced. So essentially, removing these introns is one big thing. Pardon me. And then protecting that mRNA from degradation is also very important too. There's no point, you know, going through transcription if your mRNA just gets chewed up as soon as it's produced. In addition to that, particularly in eukaryotes, these ends, the 5' and the 3' UTR, are really important for translation. 3' UTR also for RNA stability, but we're not really going to get into that. So essentially, we've got to do two big things before that mRNA is useful, right? Before it gets into the cytoplasm. And that's what RNA processing is. And then obviously, once we get that, we then have to, where are we at? Turn it into a protein, right? And so that's really, pardon me, from this part onward, or a little bit earlier, actually from about up here. So that's what this PowerPoint is really going into. The control of, or the actual process of that, producing a functional mRNA from what's called a pre-mRNA, which is very specific to eukaryotes. Prokaryotes don't do it. And we'll get into some other differences later. And then once you have that functional mRNA, then you have to translate it into protein. And that actually, even though it's really quite simple, to be honest, there's not an awful lot going on other than the initiation of translation, which looks a little crazy. But translation is like transcription. It's really quite simple other than the translation aspect of it, right? Going from nucleotide to amino acid, right? So that's really what these, what this PowerPoint is all about. Okay. And I'm going to jump around a little bit in here because this order doesn't work quite as well in person as it does when you're just kind of reading through it. So essentially there's two things that, I don't know, three things that have to happen. Let's start here. Right, so you're able to see we're on slide 9 of PowerPoint 16 right now. So what you have to do first is you have to cap the new end, right? So if you imagine, I wish I could, actually I've got a whiteboard now in case we need to use that. So shout out if you need stuff drawn out. But essentially, if you imagine that the transcription started here around where, let's say, A is in this diagram, right up here. As soon as that mRNA emerges from the RNA polymerase, the holoenzyme, the whole complex that does the work of transcription, it needs to be protected. So it has a cap stuck on it, which is kind of RNA, but it isn't recognized by RNases, RNA enzymes. Right, so that's the first thing that has to happen. The new end has to be capped, otherwise it will get broken down. Then the RNA polymerase, goes along the DNA and makes more mRNA, and at some point it's going to transcribe an intron, right? And so as soon as that happens, and that again, that kind of pre-mRNA is like flapping in the breeze outside of the enzyme, then what's called splicing machinery, or the spliceosome, right? Which is a collection of RNAs and proteins that recognizes those junctions, right? So they have particular characteristics, that bit here and that bit here in RNA. As soon as the intron emerges, it gets spliced out, right? And this is very much like editing an old film. Like you literally cut it 2 places and then you stick it back together, right? And that happens as it goes along. So if you look at this diagram at the top, right, each of those paler parts is an intron. The darker parts in the DNA are exons. And so those introns will be spliced out as they're produced. So essentially you're processing the mRNA as you're making it. It's not a make it, then process it. It's do it all as you go along, essentially, right? Which kind of makes sense in terms of efficiency. And then the very last thing that has to happen is the transcript, and this is, I think, I'm not 100% sure, I think it's part of termination transcription, termination of transcription termination, which we covered in the last PowerPoint. When that mRNA is cut, it's going to be cut in a way which allows what's called a poly-A tail to be put on the end of it. Right, and this process is called polyadenylation. Right, so you can see as it goes along, right, these introns are being spliced out, as it says here, not necessarily in the order in which they're transcribed, but as transcription is going along. And at the very end, the mRNA is cut and multiple A tail is put on the end. A good analogy would be the telomeres on the end of chromosomes, right? So the poly-A tail is like a buffer that protects the end of the mRNA from being chewed up. And there are proteins that bind to that poly-A tail called, rather unimaginatively, poly-A binding proteins. And those protect the end of that tail. They kind of mask it off so that RNases can't recognize it. But in addition, those poly-A binding proteins and this cap on the other end, the 5' cap, are also involved in the initiation of translation, right? So all of that is important for both the mRNA to hang around and also be functional. And so what you end up with at the end of that processing process, I guess, is an functional mRNA, right? It's no longer, this is a what you would call a pre-mRNA. Now that even says it here. So it's not usable in its freshly transcribed state. It has to be quite heavily processed first. But when it's finished, it looks like this, right? So you have a 5' cap, right, which is used in recruiting the ribosome to the 5' UTR. Somewhere in here, you've got your ATG, or rather AUG. Sorry, I was gonna slip up on that one. So we have the start of the reading frame or the coding sequence here. Goes all the way through, right? It's continuous. There are no introns, and there's going to be a stop codon somewhere in here, right? Then you have your 3' UTR and your poly-A tail. This, as a whole, is a functional mRNA, right? It will then be exported from the nucleus to the cytoplasm, and then you'll have a ribosome assemble on the end of this this mRNA and then kind of scooch along and start translating it. Okay, so does that make sense? We can go into the details, obviously, in a little bit, but I wanted to get the overview first. Any questions? Okay. Oh no, Alana.
I think it makes sense. I did kind of want to go over slides 7 and 8 a little bit.
Mm-hmm. Yeah, come on.
With slide 7,
Oh yeah, splicing.
there's a big picture there on the right. I'm not exactly sure where this close-up on the left is fitting into the big picture on the right.
It's the other way around.
Oh, okay.
The panel on the right that I'm circling here, this is a blow up of the junction here that forms that loop.
Okay, that makes a lot more sense.
Yeah. So essentially what it's doing is it's taking, this is the kind of the original intron structure, right? The bit along the bottom.
Mm-hmm.
And then the 5' splice site that's cut, and that end is stuck back over via the free phosphate to the 2' carbon on this nucleotide here.
Okay.
If you look kind of closer, you've got the 3' nucleotide with, sorry, nucleotide 3' carbon, sorry. The 3' carbon, which phosphates are normally attached to, but that 2' carbon is where this intron is looped back, and it is called a lariat loop, like a lasso type thing. And so that's a classic structure of an intron, and essentially what that allows, and it's a little more complex. So this is a fairly kind of simple overview, this big picture on the left, because what's actually happening is if you see here, is you're actually bringing, it is cut, right? So if we go back, let's draw this out. It's a pain in the backside to, let's see if I can, all right, I schlepped this all the way back from work on the back of my bike. Hope you made a lot of noise. All right, so this is what Jupiter and Io look like.
Can you switch to full screen so we can see a little better?
Oh yeah, good point. Yeah, there we go. All right, can you see that? Actually, you know what I'm going to do? I'm going to turn this light off too, because I get some reflection. It's a little dimmer, but hopefully. Okay, so essentially what you have is this. If I need more colors, I think I've got another couple. Right, so this is the intron here, right? And what the spliceosome does is it recognizes is it GU, AG, and then there's an A there, right? So that's essentially there's a little bit more to it than that, but that's the basic function, no, not functions, characteristics, I guess. I'm not using my words very well right here. So essentially what happens is that this part here is cut, and then it's looped over. And in the process, it brings the first exon with it. So what you end up with is, so this is exon, this is a lot harder than doing it on the whiteboard in class. All right, so you end up with essentially your lariat loop, right? And the hinge point is that A that's kind of, where is it at? This one here, right? That's the point at which that intron hinges, right, to form this loop. And so even though this is cut, it's also brought the intron 1, I'm kind of writing it quite specifically like that, making sure you don't have any reflections. The spliceosome brings that intron 1 with that kind of, with the intron essentially, even though they're not together anymore, they're cut, right? They're not actually physically attached. It brings it round so that when you cut that part of the intron off, then you have essentially, didn't plan out my board very well. You then have intron 1 and intron 2 kind of right close next to each other like that. And then you simply stick them together. Like so. Right, so actually you can see the whole thing now. So the first process is you cut at the 5' end of the intron. You loop it round using this hinge point here. That brings the now kind of is free exon 1, right? It's not really free. It's attached to the spliceosome or the SNRPs, right? And then that allows that junction down here, the 3' end, to be cut off. And there are, yeah, I'm sorry, I don't know where my other markers are, right? There are other components in here, the spliceosome. It's actually called that, I'm not making that up. Right, that then is able to ligate the free ends of, exon 1 and exon 2 together, and then you get your contiguous mRNA. That's basically what splicing is.
I had a quick question about the snRNPs.
NERPs.
Yes. Where exactly are they coming from? It said in an earlier slide that they contain sequences complementary to splice junctions, but where do these come in from?
So these are small, they're called non-coding RNAs. So they're similar to like ribosomal RNA, for example, or tRNAs. So they're produced. Let's just share my screen again so I can SNRPs. That's probably this one, yeah. So yeah, they're basically small non-coding RNAs which are produced from genes as well, right? They are encoded by genes like ribosomal RNA is. They're expressed all the time, so there's always some of them around. And then they complex with small proteins, which are produced from other genes. And I think they're all made in the nucleolus. Pretty sure that's where they're made from. That kind of digging into my memory. So they're actually made in the nucleus, and then which is also where ribosomal RNA is expressed from too. So yeah, that's where they come from. They're actually produced from DNA, and then they get assembled together just as the ribosome does, and then they're directed to wherever they need to go to do their job.
Okay.
Yeah. There's an entire universe of non-coding RNAs. Right, we've covered a few of them. SNRPs are one, ribosomal RNA and transfer RNA are another two, but there's an enormous number of non-coding RNAs that do all kinds of things. We just aren't able to get into that kind of detail here. It's also a fairly difficult concept to get because they do things even though they don't encode for protein.
Okay.
Right, which is somewhat out of the mainstream of like the central dogma and gene expression. Yeah, actually one of the really interesting things they do, where are we at? Oh, we don't have it on here. This will do. So one thing that non-coding RNAs do is they actually bind to the 3' UTR and they affect the stability of mRNA. So one way in which you can turn off gene expression is by getting rid of the poly-A binding proteins on the poly-A tail. And there are non-coding mRNAs, non-coding RNAs, which target specific mRNAs, and then that results in their degradation. So there's a whole nother world out there, right, in terms of control of gene expression. The complexity is astonishing, to be honest. So yeah, those SNRPs are just one of many non-coding RNAs that do functional stuff, even though they don't actually encode for protein.
Okay, that makes sense. Thank
Yeah.
you.
No worries. Such a cute dog. You have to show me pictures someday. Okay, so the cap is really simple. It's just that, right? It's called a 7-methylguanosine cap, or once it's been put on, it's a methylguanine. This is at the 5' end. The poly A tail is also crab sticks. Here we go. Pretty simple. Let's put this back up over here. Right. Oh, who's that? Ah, it's my wife. Morning, love. It's just a consensus sequence in the pre-RNA which directs an enzyme to cleave that pre-mRNA at this point, right? Oh yeah, there's our uRES rich sequence. So I'm pretty sure that's part of transcription termination. Again, not 100% sure, but I'm pretty sure it is. And then that free 3' end is the site of that polyadenylation, right? So capping and polyadenylation are really simple. Splicing is a lot more complex, but again, it's based on fairly straightforward principle. If there's a splice site at the 5' end and a 3' end of this intron, and a hinge point or branch point in the middle, right? And that's pretty much it. And there's a whole bunch of these U things which do the job. So I'm not going to be quizzing you on which U thing does what, right? Because I don't think that's especially interesting. Pardon me. But you do need to know that these SNRPs, or small nuclear ribonucleoproteins, form this spliceosome, right? So those are what do things, right? In terms of bending the mRNA round, cutting it, and then religating the exons back together. Okay, so that's processing. Any questions on mRNA processing in eukaryotes? If you do, just write, and you can't think of them right now, but they come to you later, just write them down and we'll go over them on Thursday. Okay, so translation is, the complexity of translation is in the initiation, right? It's probably actually no, it's in the initiation and in the actual translation, right? So what I mean by translation is, where are we at? Here. Right, so this is, some of you have seen this already because you're in my Gen Bio 1 class, or 2 of you are, let's say. And so this is the genetic code, right? Essentially what this is, and hopefully this is a recap for you really more than anything, is the code which tRNAs read in order to put the correct amino acid in the right spot. So the process of translation is taking the information of the mRNA, which is essentially a copy of the information in DNA, and using that to assemble amino acids in a sequence, polypeptide chain, in the correct order, the correct ones that should be in there. And then that polypeptide chain, that kind of long string of beads essentially, right? Then that gets kind of, assembled into the functional 3-dimensional protein. That's later, right? And so this genetic code is how that is done, right? Or rather, how you translate from these words, right, which are the codons, right, the 3-letter words that constitute the genetic code, how those are then translated into amino acids, right? UCU into serine, GGU into glycine, for example, right? Or stop, right? The ones in red are stop codons. And so that's that's the extra wrinkle, right? And this genetic code, right, the specificity of translation, or really how well it occurs, is all due to, where is it at? These things, right? So these are another example of a non-coding RNA, Alana, and they are essentially a structural RNA, right? So they form a particular structure and they have a very special shape. We're always drawn in 2D as these like cloverleaf patterns, but really they look like, they're kind of like Santa's hats, essentially, upside down Santa hats, in my opinion. And they're a good example of how RNA can base pair and form structures, right? They're an excellent example actually of that. But the two key points is down here, this part called the anticodon, and this part up here, which is where the amino acid gets attached, right? So tRNAs, when they're charged, when they have an amino acid, that amino acid is going to be stuck up here, kind of around where I'm circling on that slide. Now the specificity comes from the enzyme that does that, which is, let's see if I can remember, amino acyl tRNA transferase, I think. Hang on, what is it? Oh no, amino acid tRNA synthetase. There you go. I always get that last bit wrong. And so what they do, and you can see it down here, is they have a pocket that fits the tRNA and the amino acid, right? The amino acid is stuck there using ATP. That tRNA fits that pocket, and really, really importantly, you see at the bottom, this, it's kind of small on your screen probably, that anticodon down here, that is what is recognized by this enzyme. So this enzyme uses that anticodon to determine whether or not it should add that amino acid to the other end of the tRNA. Does that make sense?
I think it does.
Cool. Sounds like, I don't know if you ever watched, is it Pete's Dragon? Kind of imagine that you're like sitting in like a cave with this giant dragon like snoring behind you or something.
No, it's so funny. I was on the phone with my mom one time and she asked me what grown man was sleeping in my bed.
Funny. Just your dog. Okay, so that's where this specificity comes in from. It's from the anticodon at the end, right? And anticodon means exactly what you think it does. It's the opposite of the codon. So if you have, let's say, AUG, which is a start codon for methionine, then the anticodon would be the opposite of that. So it would be UAC, right? Because U matches A matches U, and C matches G, right? And that anticodon is what's used to stick methionine, or would that be UAC anticodon is what's used to stick the methionine amino acid on the other end. So that's actually one of the tricky concepts, I think, to get for translation. This is really what makes it all happen. The rest of it is really just like the nuts and bolts, the mechanism, essentially. The other part is the initiation of translation, right? So this varies between eukaryotes and prokaryotes. There are several differences actually between the two, but at least so this is a generalization for eukaryotes. So this is your mRNA up at the top. You've got your 5' UTR, your 3' UTR, your poly A tail, and then you have your coding sequence, or CDS, or open reading frame. Right? There are lots of different words for it. which is defined by the start codon and the stop codon. Right, so this is where translation will begin, this is where translation will end. And so in terms of initiation, essentially what you have are poly-A binding proteins, right? So that's the purple kind of circles. And then you've got cap binding proteins, and those recruit the ribosome, or at least part of the ribosome, to the 5' UTR of the mRNA. And so in terms of prokaryotes versus eukaryotes, prokaryotes are pretty simple. Let's do prokaryotes first, actually. Easier to put the simple stuff first. Prokaryotes simply use an inherent sequence in the 5' UTR. It's called a Shine-Dalgarno sequence. Actually, I remember that from my undergraduate times, which is kind of crazy. And what that does is it recruits the small subunit. So all ribosomes are both prokaryotic and eukaryotic, are made up of a small and a large subunit. The small subunit is involved in mRNA binding. The large subunit is what does the catalyzes the peptide bond, essentially, right? So both prokaryotes and eukaryotes, the small subunit always binds first. The difference is in the prokaryotes, that small subunit binds directly to the start codon. So you can see the start codon here, that AUG. The Shine-Dalgarno sequence makes that small subunit bind specifically to the start codon, right? And that other bit stops the large subunit binding. And the reason why is because you need your methionine tRNA to come in first. So methionine tRNA comes in, binds to that start codon, and then that is what is used to bring in the large subunit and, you know, burning some energy too. So in prokaryotes, the small subunit binds to the start codon. The initiation tRNA comes in, and then the large subunit binds. Right, so it's a very sequential assembly of those components. The difference in eukaryotes, hey, get away from there. The same kind of process applies, right? We're going to have our small subunit come in, but in this case, this all begins right at the very end, right? So you can see we're on slide 19 here. Our start codon, our AUG, is over here, right? It's way off to the right. It's not where the ribosome is going to bind first. So now we have a bunch of called IFs, sorry, or E is for eukaryote, I think. IF is initiation factor, right? So these initiation factors come in and they bind to that 5' cap. That's how they know where the end of the mRNA is, right? It's that it's like the signature, essentially. In doing so, then they can bring in both a small subunit, and there's a whole ton of other stuff as well, which we're not going to talk about, and a initiated tRNA, right? So this is our methionine tRNA, and you can see here, you know, I wasn't, you know, joking. Its anticodon is UAC, right? And that UAC, in a little bit, not right now, is going to recognize this AUG. So that all assembles at the 5' end in the 5' UTR. And then what it does is it scans, right? Essentially, it's going to go along the 5' UTR until it finds that AUG, that start codon. Oh, sorry. Didn't sleep very well last night. As soon as it does, then the large subunit comes in, right? So this big boy comes in and binds so that that first tRNA, the methionine tRNA, is in the peptide bond forming site. and that's pretty much it, really. And then as soon as that happens, then it's free to start processing along further, and a tRNA for, this is for phenylalanine. I don't actually know the genetic code, all of it. That then comes into the A site, which I call the access site, but it's actually called the amino something site. Yeah, amino acyl tRNA binding site. Access is a lot easier to remember. Right, so that phenylalanine tRNA comes in, the AAA anticodon on the end of it recognizes the U codon of phenylalanine. It's not the only one that encodes phenylalanine. Let's see. PPPPHE. Can I actually see? Oh, here we go. There's two. Let me double check. Sometimes they pop up somewhere else as well. No, that's it. So you'd get the same. Let's go back up here. You'd get the same amino acid coming in if this was UUU or UUC. However, the tRNA would be different, potentially. Right, so it could be a AA A or an AAG anticodon. Sometimes you can get away with that third one not quite matching right. That's why the third base has so much what they call wobble, right, in terms of variation. Anyway, as soon as that phenylalanine tRNA comes in, then a peptide bond is formed between the methionine and the phenylalanine, right, the amino acids, and then it processes one more over, and it keeps doing that and cutting off those tRNAs as it goes, and then that empty or uncharged tRNA then leaves through the E site or the exit site. And that's pretty much translation until we get to the end. And the end is really dictated by that stop codon. Right, so you can see here, actually, if we go back to the genetic code, there's three of them, UAA, UAG, and UGA. It's not so much that these code for stop, it's that they don't code for an amino acid. So when you get to that point, right, you can see one up here. What does that say? That's UGA, right? So that's one of the three. When that happens, this ribosome is going to, again, process along one codon, but now you're going to have a stop codon in the A site. There's no tRNA that's going to be coming in there, right? And so there's not going to be any extra amino acid added, right? So now you can actually see, in terms of mutation, the introduction of stop codons is really bad news, because it doesn't matter if there is actually the rest of the peptide sequence or the coding sequence after that, it's just going to stop, right? You can't skip over a stop codon. They are very definitive. And so once that stop codon is in the A site, now what we have is what's called a release factor. And that release factor essentially results in the breaking of this bond here via GTP or GTP hydrolysis rather, and the dissociation of the ribosome. So it kind of comes in and it levers the ribosome apart so that the large subunit and the small subunit both fall off. And that's the termination of transcription. And that's the same in eukaryotes as it is in prokaryotes. Right, I think that's, you know what, let's leave the rest for Thursday, but have a quick think, see if there's anything that you want clarifying from this part, translation. If there's anything that doesn't make sense or you want repeating or clarifying.
I had a question. on the, what does the UTRs do exactly? Does that just help it, the RN or the small subunit bind to the mRNA, or
Yes.
that's the,
So the 5' UTR is, so essentially in this diagram, which is slide 19,
mm-hmm.
the 5' UTR here is all involved in ribosome binding.
Oh, okay.
That's the role of the 5' UTR. Pardon me. Also, the 3' poly A tail is also involved in ribosome binding and stability and stuff.
Mm-hmm.
The 3' UTR, so this part here on slide 18, that's actually involved in regulation of mRNA stability. So if you remember, I said that poly A binding proteins bind to the poly A tail and protect it. So in this 3' UTR region, there are, depending on the mRNA, right? There are non-coding RNAs called microRNAs, or there's also siRNAs. There's lots of them, small interferon RNAs, et cetera, et cetera. Those will bind to that 3' UTR, and they will force those poly A binding proteins off the poly A tail. Yeah, and then that results in the mRNA being degraded from the 3' end. So the 3' UTR is very much involved in controlling the stability of the mRNA. Yeah. Again, that's so when we talk about gene expression being DNA to mRNA to protein, it's kind of simple way of thinking about it, right? But really, there are so many different points along that process where you can change the end product, which is how much active protein you have. We're really talking about the kind of the main bits, essentially turning genes on or off, and that's pretty much it, really, what we get to cover. But there's an awful lot more, a huge amount more, actually. You know, in terms of going from DNA to functional protein, including messing around with the protein itself, right? Proteins aren't always produced in their final functional form. And controlling the stability of that mRNA transcript is part of that. Because if you think about it, if you need only a protein only produced for a very short period of time, you want to be able to turn that off very precisely, let's say in development, for example. And so as soon as that mRNA is produced, it will be translated over and over and over and over again, right? If it hangs around. So if you don't want that to happen, you have to get rid of that mRNA fairly quickly, right? It has to be stable enough to last so the protein gets produced. Otherwise, you don't get gene expression. But you don't want it hanging around forever because you don't want that protein produced forever because it will screw things up if it does. And so one way of turning off gene expression is to make those mRNA transcripts be degraded. Doggo. He's finally got out of bed. Lazy ass dog. Although I probably shouldn't say that. Alana's, I imagine, still sleeping.
She is.
So yeah, that's what the 3' UTR is all about. The 5' UTR is much more due to, or much more involved in process of translation. 3' UTR is much more involved in the stability of that mRNA.
Oh.
Any other questions? Again, if you can't think of any right now, make a note of them and bring them on Thursday. Nope. Cool. All right. So on Thursday we will. The rest isn't particularly huge. I'm just. You're probably feeling a little bit spaced out right now. There's a lot of stuff to absorb. The rest is really about some just extra wrinkles, really, and difference between eukaryotes and prokaryotes in terms of gene expression. So we'll go over that on Thursday, and then use the rest of our time on Thursday to basically go over anything that you want to go over for exam 3. Sound good? Oh, hang on, we've got a chat. What's the chat say? Come on. This is a pain. Here we go. Oh, cool. There's John. Sorry, John, I didn't notice that you joined. Awesome. All right. So she'll see you on Thursday. Any questions? You can email me them as well, or just save them for when we see each other on Thursday too. Cool. All right. Catch you later, everybody. Enjoy the rest of your week, and I'll see you on Thursday.
Alrighty, have a great day.
You too.